In our last video talking about types of transport, we went over passive transport, a type of transport that first doesn't use any energy, and second, usually abides by the concentration gradient. In this video, we're going to be talking about active transport. And again, we have to reiterate the two main differences. We said that passive transport doesn't use energy, while active transport does use energy. Does use energy. And in this case, it's in the form of ATP, adenosine triphosphate. Secondly, the active transport usually goes against the concentration gradient. And remember from last video that the concentration gradient is essentially this idea where areas of high concentration of a molecule or a substance, well, these molecules would rather go to an area with lesser amounts of this substance. But in active transport, these lesser amounts actually go to the higher amounts. And that's why ATP is used in the form of energy, to push against this concentration gradient. And in types of active transport, we have to go over the three main types of active transport. One of them actually is called active transport. Secondly, we have endocytosis. And lastly, we have exocytosis. Now, in active transport, just like passive transport forms where it uses the protein membrane to channel through substances, well, there's a protein channel. In this case, when we have this protein channel embedded within the cell membrane, we use this protein and use it, of course, with energy to push molecules against their concentration gradient. And a good example of this is the sodium-potassium pump. Sodium-potassium pump. Now in the cell, and let's just call this the inside and this is the outside of the cell. In the cell, an increase of sodium or a high amount of sodium can be really bad. And what the cell does is it actually pushes sodium outside of the cell, even though there's already a plethora of sodium ions outside of the cell, and at the same time brings in potassium ions, which actually help the cell do metabolic functions. But within the cell, of course, there's already a ton of these potassium ions. And because ions don't regularly diffuse through the cell membrane, a concept that we've explored and touched on in these past couple of videos, of course, we need a protein membrane. And since it's going against the concentration gradient, we need this ATP. Secondly, we have to talk about endocytosis. And of course, endo means inside, which means that endocytosis is the cell bringing external objects inside. So you can really envision it like this. There are two main types of endocytosis. We have phagocytosis and we have pinocytosis. The main difference between these two is just that phagocytosis usually involves the uptake of solid objects, and pinocytosis is more of liquid objects like water or other liquids. And we can envision it in something like this. We have the cell right here, this huge circle, and then we have, for example, in phagocytosis, another external object. We'll just color it blue. What actually happens is that the cell uptakes this, and it starts to look something like this, with the little wedge in between where this other object is placed. And then afterward, the cell completely surrounds it. So these parts of the cell membrane actually surround and enclose this object. And then this object gets transferred into the cell and inside of a vesicle. So now what we have is this cell with a vesicle. And in the inside, we have this blue external object. And then from that, the vesicle dissipates and the cell now contains that external object. Of course, this does take energy because creation of vessels and stuff isn't exactly something the cell can do for free. And lastly, we have exocytosis. Now, exo means outside, which means that exocytosis in the cell is taking something within the cell and pushing it to the outside. This can happen in many types of things, such as within the Golgi apparatus, in which the cell either places objects within the cell membrane or pushes it out. And it looks something like this. Now we have our purple cell and the nucleus, and then we have this other external object. This time we'll color it white. And we want to get this outside of the cell. So what happens is that this object actually transverses to the membrane. So it actually gets onto the membrane, and we see something like this. And then what happens is that as this object tries to get pushed out, as the cell tries to push it out, the object actually becomes encircled within the membrane. So it tries to push out. Now this object is in the very corner, and it's pushing out, it's creating a sort of bump. And then what happens in the cell is that this object now is enclosed within its own little vesicle. And of course, this vesicle detaches from the cell, goes external from the cell, and then releases this object. This object is now free outside of the cell. And after this basic explanation, we can kind of understand the overall arch of active transport. The use of moving objects in and out of the cell and going against concentrations can provide very useful for the cell in certain situations.